Hi, I'm Michal Grabowski from the University of Lodz. I work as an associate professor in the Department of Invertebrate Zoology and Hydrobiology. I'm the head of laboratory for biogeography and invertebrate ecology there. And basically, my key taxonomic interests are amphipods and other Malacostracan crustaceans in freshwaters and also aquatic insects. And also, I have a background in biological invasions in aquatic environment. So, actually, my main work is in evolutionary biology, and I'm particularly interested in evolution of freshwater diversity in Europe, and particularly in the Carpathian Arch and Mediterranean region, especially Balkan Peninsula, because these are the diversity and endemism hotspots in Europe. Extremely interesting. So my, let's say, barcoding interest came from my works in evolutionary biology, and it's thanks to Felipe Costa, who actually convinced me that I should pay a bit more attention to CO1, which actually showed us a very interesting patterns that I will talk about in a while. So I'm a new acquisition to Bolt, and thanks to this, I don't need to feel responsible for these shameful numbers. And if you look at this closer, then you realize that even most of this is just deposited from people from outside that just collected in Poland. So for some weird reason, DNA barcoding was completely neglected in Poland, and I don't understand why, because we have several strong groups working with molecular ecology of plants and animals, but for some reason they don't do barcoding at all. So there's one thing you need to know, that we are sharing a lot of our biodiversity with Germany, so then maybe we can say, okay, we just relax and let the Germans do everything for us. That would be okay. And now, as Belarus also joined, so they are our neighbors from the east, so they do the other part. So we can just sleep well. Okay, so you know, with such numbers, I was pretty convinced that we never had any barcoding initiative in Poland at all. But then Martin Geiger from Germany told me, oh, you know, I've heard about something that happened some time ago, and then I dug, you know, deep enough, and then I found out that in 2006 there was something that was called National Plant, Fungi and Animal DNA Bank. That was funded by our Ministry of Science and Higher Education in that time, and it was a consortium of several institutes, and the leading institute was Museum and Institute of Zoology, Polish Academy of Sciences. And then I even found some web pages in some repository, and the last update was 2008, so I think that it did not survive this year. So I think that there is no prospect for this anymore, and if we want to have any, you know, national initiative, we need to create one from scratch. All right. So, but that does not mean that nothing has been done in Poland at all. It was mostly my team that developed a project which is reference DNA barcode library for the European freshwater epigean Malacostracans. The only problem is that we are more focused on, you know, Southern Europe than on Poland. And we have a quite large number of collaborators, of which the main are the University of Burgundy in France, University of Zagreb in Croatia, University of Montenegro, Skopje, and Hydrobiological Institute in Ohrid in Macedonia, Evora University in Portugal, Natural History Museum in Berlin, and so on and so on and so on. We cooperate with all these people. This initiative is funded within several projects which are dealing mostly with evolutionary biology of crustaceans, and they are funded mostly from the Polish National Science Center that likes to give money for such large-scale initiative. And also, we are partially funded in terms of fieldwork and people exchange by the EU Coast Action DNA Aquanet, which is really a great thing. And then the fieldwork is also partially funded by the local collaborators, and then we just bring them to our lab with their collections, or they send us the collections, and we are doing the barcoding. And our target taxonomic groups among Malacostracans are amphipods, isopods, decapods, and mysids. And of course, like, there is this bigger goal than just providing the DNA reference library is just the understanding of the spatial and historical patterns of diversity. So Malacostracas are cool because there are not that many of them. There's 200 plus species in freshwaters in Europe, so that makes them pretty much coherent and easy to tackle. So that's how they look like. So and these are the places that we are usually sampled, which are very nice. So from lowlands to the high mountains, everywhere in Europe almost. So and really, so that's how it looks at this moment. So we barcoded around 9,000 individuals, and already more than 7,000 are submitted to BOLD from around 1,000 of locations all around Europe, and then you can see that particularly here we have most of them. They provided 350 new beans, and we detected huge cryptic diversity, and this is enormous because around 200 of these new beans, this is within two morphospecies, which is Gammarus phosarum and Gammarus balcanicus, because they are widespread in Europe, and it appears that they are just conglomerate of really highly divergent lineages, and they are extremely endemic to even single localities in the Carpathian Mountains or in the Dinaric Mountains in the Balkan Peninsula, and this divergence dates back to Miocene. So it's really great. So what else? So this is the group of principal collaborators. So as you can see, it's a nice crowd of young people, mostly very enthusiastic from different countries. And now this initiative attracted more and more attention, especially from the Balkan countries. So we are extending this network quite a lot. So now I'm thinking about another project which would be providing the reference library for target aquatic insects groups in Poland, particularly because, well, primarily because I'm from the department that works with aquatic life. So there is a nice network of people in Poland that also work in hydrobiology. So so they can help with collecting the materials. And the main target groups are odonates, ephemeropterans, plecopterans, and trichopterans. And also there are not that many species because it sums up to like nearly 500. So it's not so so bad. So coming to this SWOT analysis. So concerning strengths, the thing is that Poland has a very long tradition of zoological and botanical research, and we have our fauna and flora and mycoflora and very well documented. We've published checklists, catalogs, identification keys, and so on, partially available online. And there are several initiatives which are working nice for digitalization of Polish zoological collections, which is also cool. We still have a representation of well-trained taxonomists in different groups, and which are experienced in field studies, and several strong institutions, both universities and Polish Academy of Science, with modern molecular labs and some sequencing facilities funded largely from the EU grants. And I would say that, thanks to the fact that we are experiencing like stable economic development in Poland since 1989, so there are reasonable perspectives for funding, especially like more large-scale studies. But recently, with our new government, which gets famous worldwide, then I'm not that sure how it will develop, but well, for weaknesses, there is definitely the absence of the barcoding initiative at the national level. But on the other side, Poland is quite a bureaucratic country, and that's we are following Germany. I think that now we are even better in this. So many of these large initiatives and consortia, they are just drowned in formalities. So I'm not that convinced at this moment that it would be the right strategy. Maybe like step by step, combining efforts of different groups slowly would be better. Then another thing is that there are not enough early career molecular taxonomies. Basically, we are experiencing, this demographic decline in Poland, so there are not enough students, and especially students that are motivated. So this is a big problem. And of course, the studies which are like kind of faunistic or floristic, that mean identification of something that was just collected in the field. It's not well funded. I mean, you need to have really such a better goal, better for the finding energy agency, because they just don't like the taxonomy, the simple taxonomy. So for threats, this may be the funding-related issues, simply the funding instability, because the project they last usually for 3 years. So then you need to apply for another one. You never have a guarantee that you get one. And then for the opportunities, I would say that this is our collaboration with the Balkan states, and we have really good experience working with these teams, and they are very keen on joining any kind of initiatives, and also like other countries in Mediterranean basin like Greece, Tunisia, but also Ukraine and Lithuania. The only problem in these countries usually is lack of funds. So it's a big problem as a matter of fact. Okay, so I think that was all I was going to